Hello, everyone, and thank you for joining us today. I'm Sean Fensky, Editor-in-Chief of Medical Product Outsourcing and moderator for today's program. You've logged on to the MPO webinar, Irritation Testing is Becoming Less Irritating. Before we get started, I have a couple of housekeeping announcements. For those of you who are interested in the information you hear today, once the webinar has ended, or if you'd like to forward it on to a colleague, this session will be archived for a year. The archive will be able to be accessed through the Medical Product Outsourcing website, which is at www.mpomag.com. Also, we will be accepting questions throughout the presentation. You can type your question into the Ask a Question box in the webcast window. We will collect all the questions and hold them until the end, but please feel free to type in questions as you have them. And now, before I introduce you to today's presenter, I'd like to take this opportunity to thank our sponsor, Nelson Labs. Nelson Labs, a Sotera Health company, is a global provider of laboratory testing and expert advisory services. The firm performs over 700 rigorous microbiological and analytical laboratory tests across the medical device, pharmaceutical, and tissue industries. Nelson Labs recognizes that every test matters and requires solutions to complex problems to improve patient outcomes and minimize client risk. And now, let's get started. Today, we're joined by Audrey Turley, Senior Biocompatibility Expert at Nelson Labs. Audrey has 20 years of experience working in research, laboratory, and test design functions in the medical device industry. She is a biocompatibility expert, having performed all the in vitro tests offered at Nelson Labs, which include cytotoxicity, hemolysis, and genotoxicity. Audrey also has experience performing, consulting, and training on bioburden, microbial IDs, and sterility testing. She trains staff within the biocompatibility department and works on a company-wide level to troubleshoot problems. And now, beginning her presentation on irritation testing, is Audrey Turley. Audrey?
Thank you, Sean, for that introduction, and thank you for joining us today. So this is a really narrow topic of discussion for medical devices, but a lot is happening in this area, so it is worth the discussion we're going to have today. I want to start off with identifying the two standards that carry the information for irritation testing. So we have here ISO 10993-1, and this is an over-encompassing biocompatibility standard that mentions how we approach biocompatibility for medical devices, and it leads us to do irritation testing, which is contained in ISO 10993 Part 10. This standard right now currently holds testing information for irritation and skin sensitization. So at the close of this presentation, I will give an update on how these standards are changing to address the changing in testing. So I want to start off with just talking about Table A1. Or Table A, this is in the 10993-1. This is also the exact table from the FDA guidance document, which is available online. This is their guidance document for biocompatibility testing and how the FDA interprets the requirements. And I just want to point out where we're led to do irritation testing. So first, we talk about what kind of a device we have, how it contacts the body, and we start very generally with, is this a surface device or is this an implant? And then we move to more specific location within the body as we try to identify where the risks are for the patient contact. And then we isolate the time duration. Once we've identified those things, we look at the biological effects that are listed here in tabular form. And you can see that cytotoxicity, sensitization, and irritation testing are required for every single medical device on the market. Either testing or some sort of risk mitigation step here. And so that's why irritation testing is really a hot topic. Out of these three tests, cytotoxicity currently is the only in vitro method that is available and approved. And so what we're moving towards is an in vitro method for irritation. So this has a significant impact in the medical device industry and especially in animal welfare. So the presentation overview today is that I want to start by talking about where we are now, the in vivo irritation test, go through the methodology, give some history, and then report the issues that we've seen with this test over the several decades that this has been in use. And then we will go over the in vitro irritation test, where I will give a history of its development, the methodology that is used, and the applicability to determine irritation response from humans. And then I will give a standards update as well. So let's talk quickly about irritation and just our definition of it, just very high level. So irritation symptoms come in the form of redness and swelling on the skin typically. This is considered an acute and localized response. So think about if you've ever used a detergent or a soap and you said, oh, I don't, this makes me itch. That's an irritation response. What's nice about these responses when we experience them is we can wash our clothes in a different detergent, and then the response goes away. So this is not something that worsens over time or escalates. It's very short, and it's caused by damage to our keratinocytes, and that becomes important when we start to talk about the in vitro method and how we assess that. So now that we've got our baseline for irritation definition, let's go in and talk about the method here. So this is for the in vivo irritation method. This is what's laid out in ISO 10993-10. The nice thing about the in vivo irritation method that we have right now is that we can adjust this and customize the testing for the location of application of the device. So if you think about contact lenses, we can do an ocular irritation test, or if you have an eye drop or something like that. Any dental product, we can do oral exposure there as well. So it's nice that we can customize it and kind of address some of the sensitivity issues that we want to see in these different locations and different tissues. But the most commonly performed version of the irritation test is intracutaneous reactivity. So this is used for devices that have, maybe used during surgery, especially for implant devices, where it's going to have a more systemic exposure to the patient. So just to give a brief outline, of that test, I want to start with sample preparation. And I'm making this more of a topic of conversation here so that we can see when I talk about in vitro, we're going to identify where our differences are. So we start here with sample preparation, which is outlined for us in ISO 10993 Part 12. We can see here that we can test either, raw materials if we need to for screening purposes, which we have here in a test tube, or in the middle of the screen, we just had this nested cube. So we really like to test final finished devices in vivo and in the in vitro assay as well. What we do is we cut up the device if we can, and we put it in an extraction container. And then we extract it in a polar and a nonpolar solvent. So our polar solvent is typically saline, and nonpolar solvent is an oil, either sesame oil or cottonseed oil. And then the extraction conditions these are the ones that are allowed in 10993 Part 12 for irritation testing. The most favored extraction condition is the 50 degrees for 72 hours. If you lower that temperature to 37 degrees, there needs to be justification for that. And the reason here is that we want to see an exaggerated extraction. So 37 degrees seems logical because that's, basal body temperature. However, we just want to do a little bit worst case escalation and make the test as sensitive as we can. So that's why we typically choose the 50 degrees for 72 hours. This slide just gives an overview of some key points about the test. So we use 3 rabbits for each solvent. So for saline, there's 3 rabbits, and for our oil, there's 3 rabbits. We do injections of the test article on one side, and then the negative control on the other side of the spine of the rabbit. This data, after those injections are done and we observe them, it's very subjective and qualitative. So that can cause issues with reproducibility. So it's important that we point that out. Our turnaround time currently is about 4 to 5 weeks. The cost is about $2,000. And our sample requirements are 2 samples at a minimum, which need to be at least 120 centimeters squared. And so, again, I'm putting all this information out there for comparison purposes, so when we talk about in vitro, we can see where those differences and similarities lie. So I just wanted to go through some of the details on the in vivo irritation test. So the injection, like I said before, we do 5 sites on the right of the spine of the animal and 5 injection sites on the left. So what happens is just before, testing within 24 hours, the back of the rabbit is shaved, and then they go through and do these injections. Those injection sites are marked, and then those sites are observed every 24 hours over a period of 3 days, over 72 hours. And so when the technician is observing the animal at each time point, they are looking for erythema and edema, so redness and swelling. And they're scoring those injection sites for each of those reactions on a scale of 0 to 4, where 0 is that we've seen no reaction and 4 would be a severe reaction. After the 72 hours, we take all of those observation scores, add them together, and divide by 15 to get an average. And then we divide those to get an average among the 3 rabbits. And then what we do is we want to compare our test article to our negative control. And we want that difference to be 1 or less when we're looking at those end results. It is typical that we will see elevated responses with an oil injection. But again, those tend to be negated out because we also see that in the negative control as well. So here are some of the issues we have with the in vivo irritation assay. It's been reported that we have poor accuracy in prediction, and so there have been times that we haven't been able to accurately identify a known irritant through the in vivo assay. And of course, something that's really obvious is that there are physiological dissimilarities between humans and rabbits and in our skin as well. And so we're going to see different reactions between species. And then the data is subjective and qualitative. So the scoring based on a scale of 0 to 4, which is based on an interpretation of how much redness, how much swelling are we observing at the site, it can make reproducibility difficult. So if you do irritation testing at one lab with one technician, if you go to another lab and a different technician, are you going to get the same results? And if you don't, is it because that there was a reaction seen, or is it because the interpretation is a little bit different at that facility? And then there's also been reporting of under and over prediction. So there's a pretty high false positive rate with this assay that has been reported before. Not as much of a false negative, those are pretty low, but the over prediction makes it difficult, especially in the medical device industry, where it's difficult to pinpoint what could possibly be causing the irritant. And then if you do have an irritation response, do you have to go all the way back to the drawing board and reformulate your device? So we've covered the gamut really with the in vivo assay, and so now I want to introduce the in vitro irritation test. So just some key points. That this test that I'm going to talk about today uses reconstructed human epidermis. So this is abbreviated RHE. This model has also been used for chemicals. It is published in OECD 439. And it has been determined for OECD 439 specifically, where we're addressing chemicals, that this method can properly categorize United Nations globally harmonized system of classification and labeling of chemicals for a category 2, which is an irritant. So you can have a non-irritant, a category 2, which is an irritant, and then for other measuring tests that are available, there is a category 3, which would be a mild irritant. So it's been determined that this in vitro method does not distinguish between a non-irritant and a mild irritant. So we're sitting at one extreme or the other. And we'll talk about how that can still be appropriate for our uses and still be a good place to start rather than going straight to animal testing. So this slide just gives some example and a visual for you to see what we're looking at when we do tissues. So there's an in vivo tissue stain on the left of your screen, and you can compare that to the in vitro, which is on the right, where we can see the differentiation of the keratinocytes, very similar in structure between in vivo and in vitro. And then just a brief example of how the tissues are grown. So cells are added to a filter that is put inside an insert, which is the smaller circle, the smaller container. And then those tissues are actually fed underneath from the media. So it's very similar to our skin, where it's fed underneath from a blood supply and grows up. And that's exactly how the tissues work. And what they're showing here with the green is where you would apply a test sample. So this is a larger image just to show that we are seeing the differentiation in the keratinocytes. And as they divide and move up in the tissue, they do provide the different layers that we're looking for. And then we do have the stratum corneum, which does provide a barrier. So I want to talk for a minute about the tissues, because these are the most important aspect of the irritation test. This is what differentiates it in the industry for this specific use. In OECD 439, they have specified criteria for tissue manufacturers that if you're using a specific tissue, there needs to be requirements that you ensure the tissue will function properly for an irritation response. So these next few slides will go over those requirements and why they are important. So the method components here for this one is about a barrier function. So we need to make sure that we have a proper barrier, not too much, not too little, and they've established some testing criteria and parameters for each tissue. Company has decided if they're going to do an IC50 or an ET50 test and what chemical they're going to use, SDS or Triton X100. And so we can see here that they've reported their limits. So what happens now for these tissue companies is those test results for a batch of tissue are reported on your C of A. So you can ensure that they're doing quality inspection on each lot of tissue to ensure reproducibility from lot to lot. This is great assurance because we don't have that same control over the animals, right? We don't have that kind of understanding of their tissue responses or their full biological system. So this steps up another level of control that we like to have in this type of situation. Another requirement is that we need to ensure we have control over the viability. So this is to ensure that we have the right amount of cells present in the tissue. So if we have too many cells, then our viability results get skewed. If we have too little, again, they're skewed the other way. And so we want to have consistency. So when we run repeated tests, we can compare those results. So you can see the tissue manufacturers here have also published their known levels. So I want to take this moment to just talk about these individual tissue manufacturers here that are listed because there's 4. And these are separate tissue brands in particular. So we have Episkin and Skin Ethic. So they're not right next to each other, but these come from L'Oreal's lab in France. And so they are different in their diameter of tissues that they offer. And that's a little complex for our discussion today and not an issue, but they just have 2 different types of products they've offered for the cosmetic industry for years. And because there is some familiarity already with both tissues, they continue to market both as they will meet needs for chemicals, cosmetics, and now for medical devices. The epiderm tissue here on the second line, that's manufactured by Mattek, and they have a facility in Slovakia and one in the U.S. in Boston. And then the lab site tissue is manufactured, I believe, in Japan. And so we have these 4 manufacturers that are allowed. And so what we want to see, if we have other manufacturers that decide to come forth and put a product on the market, that they are measuring these same conditions and same controls across the board. Okay, so moving on to morphology. So it's important that we have, those stained tissue slides so we can understand that there is the appropriate differentiation of the cells and we are getting the layers represented for the keratinocytes. Okay, now to talk about the specific test method. So this is a broad overview again, and this is for the in vitro irritation test. So this one will require 15 tissues, and I have pictures of this in the next slide so you'll be clear on what that means as far as tissues. But this is to show our replicates mainly here. So we're going to do 3 positive tissues, so 3 replicates for our positive. Our negative and our test articles look like they're more, but we're also doing 2 solvents again. We're doing a polar and a nonpolar solvent. And so we have 3 replicates for each solvent for the negative and for the test article. Our data here is quantitative, so lends to reproducibility across labs and within labs as well. Turnaround time is still about 4 to 5 weeks, and the cost is a little over or just a little under $3,000. Now, the turnaround time is established currently at 4 to 5 weeks because it takes 3 weeks to grow the tissue, and because this test is up and coming and isn't fully accepted yet by regulatory agencies, we still wait until once we receive samples, then we will order the tissues. So there's this wait time while the tissue is grown. But I do anticipate as this method gains acceptance and moves into the standards that turnaround time will be reduced. And of course, the cost is based on the number of tissues. And if you are sending in multiple samples or things can be batched, there is some ability to combine the tissues where you aren't having, this cost multiplied by the additional samples you send in. So there's some interpretation here for the turnaround time and the cost is the point I want to drive, that is going to change in the future. And then the sample requirements, again, is for 2 samples because we are doing a polar and a nonpolar solvent. So we'll go through this like we did for the in vivo. So our extraction is very similar. It's identical, actually, to the in vitro or to the in vivo method. We still want to do, your final finished device if that's what's available. We'll put that in an extraction container, do a polar and a nonpolar solvent, and then preferably use the 50 degrees for 72 hours. The exposure. So this is where we take that extract after it's been extracted in the incubator and we put it on the tissues. So that is applied topically. And I wanted to spend a minute just talking about the tissues here. So you can see that we have these small inserts. And I don't know if you can do it now while it's live on the web, but these slides will be available after the webinar. And you can blow up this image and you can see actual texture on the tissues. If you love science, you may geek out about that. I think it's very fascinating. And so what we do is we take 100 microliters of the extract and we apply it right to the top of this tissue in the inner insert. And then that is incubated over a period of 24 hours, 18 to 24 hours, depending on the specific tissue that we're using. After that incubation, we then expose the tissues to MTT. So I have an image of that here. So the tissues are exposed to MTT for about 3 hours, and then they are rinsed, and then we extract the tissues in IPA. So let's talk for a minute about what happens when we extract with MTT. So you may all be saying, well, MTT is for cytotoxicity tests. What's going on? MTT is a dye, right? So we use this frequently in cell biology. So it converts. We use it to tell which cells are alive. So live cells will convert the MTT to a formazan salt, creating this purple color. So we can see here that we've seen some live cells and some tissues that are not dead. So purple indicates live, clear indicates dead tissue. We can see on our 3rd row down of tissue that we have some spots of live tissue. So we are getting some viability, but definitely a significant amount of cell death. So what we do from this point is we take that IPA and we plate it on into a 96 well plate, and we read those color changes on a spectrophotometer. And so on this slide, we can see, those results calculated from an Excel spreadsheet. So we run this calculation about percent viability, where we're taking our optical density, so our color change from either our positive control or our test article, and we're going to compare that to our optical density of our negative control. So that negative control really becomes our baseline here. And then we multiply it by 100 to get a percent. So we can see in the data that's on the right side, on the left side of the screen, where we have our positive control, our optical density is measured, which is very low because these are our clear cells. Our percent viability is 3.4, and our category is irritant, I for irritant. And then if we go over to the chart, the graph, we see that our positive control is coming out very low. And we can interpret that all the way across. What we look at, our category here is that our negative controls, our test articles came out as non-irritants. On the chart, I don't know if it's coming up very clear, but on the 50% line, that line is red. That's our indicator of where we're crossing between irritant or non-irritant status. So this has been established by the testing performed, and that is outlined in OECD 439, where if you are below 50% viability, so you had a significant amount of cell death, that we are saying that that's an irritant, category 2 irritant. If you are greater than 50% cell growth, then we would say that you are a non-irritant. The product is a non-irritant. So one thing you may be asking is, what happens if you sit right around that 50%? What if you're 48% or 51%? Is that enough difference to really be comfortable with the categorization? So what we recommend is if you're within plus or minus 5%, so 45 to 55%, that additional testing would be a good idea, and possibly, depending on the risk of the device, we might recommend that you move forward with some in vivo testing to confirm the result. So, as much as we want to replace irritation testing for in vivo, we may still have to go there. So this is just looking at a huge reduction for irritation testing at this point. Okay, so I wanted to take a minute and just talk about MTT, because this has been our biggest question as an industry as we've moved forward with this, that there's this thought that MTT is only for cytotoxicity testing because it's used so heavily. But it's important that we want to make sure we understand that MTT is a chemical that is used to measure cell viability. So that can happen in multiple types of cell systems. What makes this different is the reconstructed human epidermis. That we're moving, when we think about cytotoxicity testing, that's just on a very basic cellular level. When we add complexity to the system, we've allowed for complexity in the response. And so we can still measure cellular functions of the RHE when we take in appropriate controls, as we outlined initially from OECD 439. I tried to anticipate some questions that might be coming in. If you still have questions about MTT afterwards, please feel free to post those. Okay. So now let's talk about changes that are happening in the standards. So as you know, as I said at the beginning, irritation testing is all documented in ISO 10993 Part 10. So what I want to talk about quickly before we go into, what's exactly going to happen in the standards is, what's happened to bring the change in the standards. So we meet for these meetings on an American level. That's the ANSI document. I'm just going to flip back to that. This document is for the American National Standard, which is also through AMI. So these are our American organizations to promote these standards. And then we also work at an international level. That's the ISO level. And so what has happened over the last 4 years is we have worked together as an industry to pull together and do round-robin testing. So this means that we've agreed internationally between many countries around the world on a standard method that we're going to do, and that agreement is also based on research that a few companies have done in their own house before they put up the protocol. So we agreed to a protocol. We also include multiple models for the reconstructed human epidermis. There was some comparison in vivo testing done as well with the rabbit test and then with human patch testing. And then we demonstrated reproducibility in over 19 labs worldwide. And this has been such a popular round-robin test that other labs are still coming on board in other countries to get the testing going as well. So with that background information to know what's driving that change in the standards, we move to the discussion about what's actually happening. So ISO 10993 Part 10 is going to remain as only a standard for sensitization tests. So we don't see a lot of changes coming for the in vivo options, but we are starting to now turn our attention as an industry to in vitro options for sensitization tests. Those options will then be laid out in the annex of this standard when it's newly released. And then for the irritation tests, we are creating a new standard which will be made as ISO 10993-23. And that will cover all irritation tests in vivo and in vitro. So for the in vivo, we don't see a lot of change coming from the animal perspective of the testing. And for in vitro, we'll put out the principle of the irritation tests, and then in the appendices, we will add the specific protocols for each tissue. So one thing that's great about this method is because we always have a hard time sharing our methods in the industry because we want to protect IP for the companies that invest in that development. The great thing here is the IP is sitting with the tissue manufacturers who want to sell their tissues so we can use it this way. So it's in their best interest to release the test methods along with the tissues as we purchase those for use in the testing. So we're really excited about this nice sharing of information that's been able to happen in the industry. So for the standards, though, that's where we sit now. We're working on writing -23. It's still in draft form, and I don't have an anticipated publication date at this point. We are meeting again in Berlin in December, where further work will be done. So I would keep your eyes out for this publication so that you know when these changes will be in effect, that you know they're coming down the road. So one other update to give is about regulatory approval. So this is a little bit of a not clear path, I guess I would say, for many countries. Typically, regulatory agencies will accept the methods once they're in the standards. But from a U.S. perspective, we didn't want to wait for that acceptance. And so the U.S. FDA has this great program called Medical Device Development Tools. And this quote here is from their website on it, where they're promoting, really having access to any type of tool that medical device companies may have to assess their specific device. So I also want to take this opportunity to say this effort that's happening, from the R&D testing, the change in the standards, the push to the FDA is all due to so many people who have put in many, many years of hard work into this industry, and specifically for the U.S. FDA's application into this program. It was Dr. Kelly Coleman who's done this. And so great hard work from these leaders in the industry. And I've got some information at the end of the presentation where you guys can have access to their work. But I wanted to take the opportunity to give credit where credit is due. So with the US FDA MDDT program, Dr. Coleman submitted the full application on September 21st, and there's a 60-day review period. I would encourage anyone who's curious to check out FDA's website. They've got some great introductory information in there, and see if there's some tool you would like to also promote and push through the program. I know the FDA is really excited about people using this for test method development as well. So we are hoping for an early Christmas present that the FDA will accept this. before the end of the year, and we can publish that. But right now, we're just waiting on their approval and their comments on the application. And I thought it would be nice to really explain how this is going to be a game changer in the industry and put some numbers up here. So this is data that I gathered from the FDA. This is the number of 510K applications that were submitted just in the first quarter of this year. So that's over 1,600 device applications. When we think about those devices, it's important to note that for each one of these products, there's a minimum of those big three tests, right? Cytotoxicity, sensitization, and irritation. So when we talk about that, how many animals, that's a minimum of like 26 animals when we think of sensitization and irritation testing. So we're making some assumptions here, but I think there's some pretty safe assumptions that this is a minimum number of animals that are used. When we look at that in terms of the number of applications received, that's over 41,000 animals used just in the first quarter, just in the U.S., and only for 510K applications. With the introduction of an in vitro irritation model, there's an opportunity to reduce that number by 9,648 animals. So that's a big impact that we're really excited about. So I would just encourage you to think about your irritation testing program at your work, at your company. If you have the option to do an alternative method even before acceptance with the FDA, if you want to start that conversation early with them and be a pioneer for your company, that's also a very good option. And we have helped companies do that with the FDA and with European notified bodies as well. So that's a great impact, and we're really excited about it. On this last slide here, I've put in the papers that are published. This contains all the work and data gathered from the round-robin testing. So you can see there are experts from all over the globe represented in this writing. Most of them are open source. I believe there's only one that hasn't been available open source yet, but I think is slated to be so soon. So I encourage you to, take these documents and read them and really pursue an alternative irritation testing program at your facility. And I will turn the time over to Sean now for questions.
Great. Well, thank you, Audrey, for that thorough and informative presentation. Once again, to everyone listening, don't forget that this webinar will be archived on mpomag.com for 1 year. Now, as Audrey mentioned, we'll have time for the Q&A session. We'll try to get to as many questions as possible. Please enter your questions into the Ask a Question box or the question box in your web browser. The first question is a two-part question. The first is with regard to in vitro irritation. Is it pH or, and I apologize for butchering this, osmolality sensitive? And then the second part is, what about particles within the extract?
Okay, thanks, Sean. I think the word we're looking for is osmolality sensitive.
Thank you.
Yeah. So I would say that as far as being pH sensitive, to answer the first part of the first question, when we did the research and development for the protocol, we did test acidic compounds in the extract, and we did not see, other than that they did, of course, cause irritation. But beyond that, I have not done any personal research about pH in specifics, but we would expect if it has a low pH, so to be acidic, we would expect that to cause an irritation response. So in that regard, we would expect it to be sensitive so that it can measure irritation from that type of mechanism in the chemical. As far as osmolality, I do not have the answer to that. So I'd encourage you to either send me an email with that question, and I can reach out to some of the tissue experts and get that question answered for you. And then the second part of that question, can you read that to me again, Sean? I don't remember that.
Sure. It was, what about particles within the extract?
Okay, great. Thank you. Let's see. We didn't see any particles in our R&D as far as the extracts, and I don't know if they would have an impact. I gather if they were large and damaged the tissue physically from that aspect, there would be a problem. But I don't anticipate, especially because a lot of cosmetics have been done here, so we're thinking creams and powders have been tested on this specific, especially with the episkin and skin-ethic tissues. I know that they've done that in the past, and they've been still able to get the results that they are looking for. So I don't know that small particles will be an issue. Larger particles, if they cause, like I said, physical damage to the tissue, I would anticipate that causing a problem. But again, an area we didn't cover in our research.
Okay, great. Now the next question: what do you have available which mimics mucosal tissue, specifically GYN tissue?
Okay, for the mucosal tissue applications, I know that MatTek in particular, I'm a little more familiar with their offerings, that they have some different tissues in development. And you can go to MatTek's website, and how to spell their company name is MATTEK. And if you go to their website, they will list all the different tissue offerings they have now. I remember when I did training there, they were really excited because they had an esophageal tissue that actually secreted mucus. So they've really had a lot of advancement in their growth of this tissue and been able to mimic human tissue environments very closely. So I would encourage you to go to the tissue manufacturer websites to see what their current offerings are for the different tissues.
Okay, thanks. The next question is: in vivo irritation testing still appropriate or accepted?
Okay, that's a great question. So the in vivo testing is appropriate, especially now because currently because these in vitro options are not written into the standards. And you can see from our discussion that there will be some point where in vivo testing will be an option and where you may want to go to after doing in vitro. So especially where we see, a borderline result, or if you do see that the sample is coming as an irritant, maybe 30% or something like that, and you don't want to go back and do reformulation and you'd just like some additional testing, we would move into in vivo at that point. So the short answer to that is yes, it's still appropriate, especially for now, and still will remain valid even after the in vitro method comes out in the standards.
Okay. The next question: can you kindly show us an example of a report that comes back to the customer so we know how to read it? Now, obviously, we can't show it, but maybe you can explain that?
Sure. Actually, I will go back to the slide where I talk about the results that are put out after we do the testing. So you will see something very similar to this graph and these results reported in the middle of the screen. So you will see the samples identified, optical density, where you have your mean and your standard deviation, and your percent viability, where you have a mean and a standard deviation. So what's important here is that the laboratory should have specified standard deviations that are allowed for especially the percent viability. And this is where we want to make sure that each tissue is responding the same. So I'm going to focus on this column for just a minute, the percent viability, where we have the mean, and we start at 3.4% with a positive control. That right there is an average of 3 tissues, or excuse me, I think in this case it was 2 tissues, but the average of the number of tissues that were exposed to the test article, in this case the positive control, and then we take all of those and then, we go from the tissue to the IPA extract of the MTT. So we actually are averaging multiple replicates of reads here. And so we want to make sure that we're not seeing anything greater than an 18% standard deviation. And that will be listed in the final report for the acceptance criteria and will be in the protocol for the test to be performed. So that's an important column to pay attention to. And of course, this category here where it has I or NI, so irritant or non-irritant. And so those are going to be key columns for customers who are getting the reports to pay attention to. And of course, a very nice visual is the graph. And that one I would pay special attention to if you're sitting above that or close to that 50%. I think a visual there is sometimes a little bit quicker interpretation. So that would be an example of what we would demonstrate in a final report.
Okay, perfect. The next question, does the U.S. EPA recognize any in vitro sensitization assay method for product testing under TSCA or FIFRA?
You know, that question I'm not prepared to answer. I did not look into in vitro sensitization assays at this point. Where I sit from where I've had any input on on the in vitro sensitization is that there are many, many methods that are out there, and I have yet to hear of one that is fully approved, or just the golden child, if we want to say that, of that we're going to follow. And so I don't know specifically about the US EPA and their requirements for that.
Okay, fair enough. The next question, when will the method be accepted by regulatory bodies?
So what we're hoping to hear back from the FDA in the next 60 days is that there is some acceptance. We've answered a lot of questions. I know Dr. Coleman has been in contact with them, and he's answered a lot of questions up until this point. So we're in conversation with them. I wouldn't anticipate anything being released before December from the FDA's perspective. As far as internationally, we have had acceptance of this through notified bodies in Europe. Europe has been really the leader in pushing in vitro assays right now to reduce our animal testing. And so we have had some low-risk products that have used this test and been accepted, but it is on an individual submission basis and not an overall notified body approval.
Okay. And another question, when will the new standard for irritation testing be published?
Yeah, so we don't have a firm date on that. We know we're continuing to work on our writing progress to separate out the two standards and make sure our language is consistent and that we have fully removed sensitization and irritation testing from each other. We will meet again in December in Berlin and address and set some dates at that point.
Okay, and I think that was actually our last question from the audience, so we'll wrap up for today's presentation. I'd like to thank our speaker, Audrey Turley, for being a part of this webcast. I'd also like to thank our sponsor, Nelson Labs, for its support. Finally, I'd like to thank you, the viewer, for attending this session. I hope you found it to be a valuable experience.